Hi everybody, in the previous video we talked about the application of the gels for cell microencapsulation in order to treat the diseases, and we mentioned, for example, treating the diabetes in which the pancreatic islet cells are not performing well and correctly inside the patient's body. So we will extract them from an animal source and implant them into the body of the patient, but the problem is that because they are from the animal source, the body of the patient will reject them. So what we have to do is to encapsulate them inside the matrix of the gel in order to prevent their direct contact of the cells with the antibodies and the inflammatory cells. And we mentioned that this matrix is a semi-permeable one in a way that it allows the diffusion of the nutrients and the insulin which is produced and also the waste products inside and outside this matrix. But on the other hand, for the antibodies and the inflammatory cells, because they are larger, they cannot diffuse inside the matrix, so they will not be removed by the body. But what is the best gel that can be used for the cell encapsulation? Well, the calcium alginate is known as the gold standard for encapsulating the cells. And what is alginate? Alginate is an anionic polysaccharide which contains the glucuronic acid and the muronic acids, and they are negatively charged. In the presence of a cation such as the sodium, only one polysaccharide will be connected to this sodium because it is a monovalent cation. But if we introduce the calcium as a divalent cation, the self-assembly of these polysaccharides will form the gel. You see here we have the egg boxes. This is the self-assembly of the polysaccharides of alginate, and you can consider the calcium as the egg which is inside that. Let's see the mechanism in which the calcium alginate is formed. Just consider we have a suspension of the cells, and here we have the polysaccharides, and here we have the polysaccharides, and we mix them together. We know that if we introduce the calcium, the alginate will self-assemble into the gel form. So we will inject this mixture inside the bath of the calcium chloride. So when it is injected, due to the presence of the calcium, the self-assembly will occur and the gels will be formed. But just consider that the formation of the gels here is not due to the increase in the temperature. It is just due to the presence of the calcium which will self-assemble the alginate polysaccharides and form the gel. So you see here when the gel is formed, the problem of losing the matrix is still present. If you remember, we mentioned before in the previous videos that some of the polymers of the gel can diffuse out, and when the concentration is decreased, we will revert back into the liquid phase. So we don't want it to happen. What we can't do is to introduce on this negatively charged gel a positively charged layer of the polycation. So we introduce this positively charged layer, but the problem is that it is not something favorable inside the body, because if you remember about the protein corona, we mentioned that when the proteins are absorbed on the surface of the nanoparticles, it, let's say, triggers the inflammation, and on the other hand, this positive charge can interact strongly with the negative charge of the cells around, and a formation of a tissue will be around it. So this is not something favorable. And what we can do is that we can introduce another layer of alginate and is positively charged to maintain the stability and the compatibility of our gels. Another thing that we can do is that we can introduce an acid inside that to remove the alginate, to remove the calcium. And when the calcium is removed, the self-assembly will remove, and we will revert back into this mixture of the cells and the alginate. So inside that we have the liquid phase, and we have the outer layer of the gel. So it forms and it acts just like a microcapsule. Okay, in the next video we will be talking about different types of encapsulators.